Okay, question 2, diagram 2.1 shows the plasma membrane structure involved in substance movement control. Okay, so name structure L and M. Okay, so structure L here, as you can see, it is known as phospholipid bilayer. And then if we look at the detailed structure of phospholipid bilayer, we have what we call as hydrophilic head, meaning that this is water loving, and hydrophobic tail, meaning that it is not water friendly. So that's why it stays on the inside of the membrane. And then we have structure M here, which known as carrier protein, and also responsible for the passage for molecules to pass through the membrane. And then number 2, state the characteristic and example of molecule that can move across structure L. So just now we know that structure L is phospholipid bilayer. So any molecules that can freely transported across the plasma membrane, it is due to its hydrophobic core, and the membrane itself is highly permeable to the non-polar molecule or lipid solute molecule and uncharged molecule. Hence, it able to pass through the cell membrane down their concentration gradient by simple diffusion. For example, we have fatty acid, vitamin A, D or vitamin A, D, E, K, oxygen and carbon dioxide. Okay, and then B, based on the diagram 2.1, state two differences between process X and process Y. Okay, so process X is what we call as active transport, and process Y is known as passive transport. Okay, so in process X, where it requires ATP or energy, but in process Y, there is no requirement for ATP or energy to let the molecules to be transported into the cell. And then number 2, the molecules from low concentration area at the extracellular environment are transported across the cell membrane into the cell. While in process Y, the molecules are from high concentration area are being transported across the cell membrane into the cell. Okay, and then based on this diagram, explain how substance A can be transported across the plasma membrane into the cell. Okay, basically just now we discussed that process Y is the passive transport, okay, where it doesn't require any energy to let the molecules to pass through the carrier protein, where substance A will bind to active site on the carrier protein. So then carrier protein will change its shape, and the changes of its shape will cause the substance A to be taken into the cell. And this process is what we call as facilitated diffusion. Okay, move into question C. Diagram 2.2 shows the mustard green stem that is cut longitudinally into strip, where the strip is immersed in distilled water for 30 minutes. Okay, and then draw the condition of the strip after 30 minutes immersed in distilled water in the box provided. So the green stem will be bent outward. Okay, it will bend outward, where this part is the cut surface part, and here is the epidermis area. Okay, so the cut surface will bend outward, and distilled water, just now I said that distilled water it is hypotonic to the cell sap of the strip. So water will diffuse into the cut surface via osmosis. Okay, remember in question 1, we discussed that in vacuole containing cell sap, they're able to absorb the water content into the cell. So water will diffuse into the cut surface via osmosis and make the cell become turgid or bloated.